So the process of generating and maintaining a normal organism over its lifetime is one that's wrought with excess. There are millions of cells that are generated, but in order to properly shape the organism, these cells unceremoniously undergo apoptosis or other types of programmed cell death. Now, it's important to realize that these cells just don't disappear, and they are actively cleared by phagocytes in the developing embryo as well as the adult organism over time. So we know that this process is an active process, and it's termed efferocytosis, and it's composed broadly of 4 steps. The first step is the release of find me signals by the dying cells, and this can include release of ATP or CX3CL1, and these molecules act as chemoattractants to recruit phagocytes to the sites of cell death within tissue. And there's been a lot of questions and controversy about what the exact role of find me signals are, because many times the systemic or serum concentrations of these molecules are higher than what is released by apoptotic cells. So the question is, are these chemoattractants and find me signals really acting locally, or are they having some other type of role? And we're starting to see new evidence now that find me signals not only act as chemoattractants, but also are important for priming the phagocytes for their future meal of apoptotic cells. The second step is also an active process, and it's the exposure of eat me signals on the surface of apoptotic cells. And this is an active process in the sense that during apoptosis, molecules that are normally confined to inside of a healthy cell, such as phosphatidylserine, are actively exposed or flipped to the surface of apoptotic cells, and these allow phagocytes to recognize these cells as dying cells that need to be cleared. Now, phagocytes do this because they employ a variety of receptors that are specific for these eat me signals, such as phosphatidylserine receptors, like the TIM4 family, stabilin-2, or BY1, which have very specific tissue expression patterns, which can explain why there's such a multitude of these kinds of receptors expressed by phagocytes. But there's also bridging molecules, such as MFGE8 or Gas6, that also recognize phosphatidylserine or other eat me signals, and then they link up with integrins or other types of phagocytic receptors such as MERTK on the phagocyte. Now, engagement of these phosphatidylserine or other eat me receptors is important for step 3, which is the actual engulfment and phagocytosis of the dead cell. Now, once inside the cell, step 4 can occur, and this is degradation and processing and directing of the immune response to these dead cells. When you think about the billions of cells that undergo apoptosis every day, as part of a normal homeostatic process, you don't want to have a robust immune response to these. You want it to be immunologically silent, and you want your response to be immunologically tolerant, which is what phagocytes have evolved over time to do. Once they have basically doubled their intracellular contents of lipids and proteins and molecules, they must somehow deal with that sort of metabolic burden, and they do that through activating specific receptors such as the PPAR gamma family or LXR, which are designed to downregulate and promote an downregulate the immune response and promote an anti-inflammatory response. Now, one thing we know, and we've recently discovered, is that a new pathway that's been described called LC3-associated phagocytosis is very important for modulating this immune response and degrading the engulfed dead cell. This pathway, termed LAP, results in the recruitment of specific members of the autophagy machinery to the dead cell containing phagosome, and we've recently identified a protein termed Rubicon that we know is now required for LAP, but completely dispensable for canonical autophagy. And this is really interesting because we know that in the absence of the LAP pathway, you get a skewing from this normal anti-inflammatory response to an increased pro-inflammatory response. And this really raises the question of what the autophagy machinery is doing in this pathway, because we've known for years that proteins such as ATG5 or Beclin, which are required for regular canonical autophagy as well as LAP, are often defective, or there are SNPs present in human patients that have autoimmune disorders such as lupus or rheumatoid arthritis or ulcerative colitis. And so the question now is, are we seeing defects in canonical autophagy in these patients, or what we actually seeing is a defect in the ability to degrade dead cells via the LAP pathway? And still there's lots of questions about how this pathway is modulating the immune response. We know how important it is to maintain this anti-inflammatory environment, but there's so many pathological situations where this must occur, where you might want inflammation or you might want some sort of response, for example, the tumor-associated macrophage, which would constantly be seeing dead cells, you would want some sort of inflammatory response in a way to recruit in maybe more T cells to help, but still this area of work is yet to be discovered. So I think we're at a really exciting time now to start looking at the role of efferocytosis and specifically the role of the autophagy machinery in terms of maintaining this immune environment that we need for homeostatic conditions.